Okay, guys, we just have a short section left to talk about in chapter 9, and it's entitled The Genetics of Animal Viruses. So this should be a short and sweet lecture. I would expect no more than about 10 or 15 minutes. Okay, so last we left, we were discussing transposons, if you recall. So this last section in chapter 9 talks about some stuff that we've actually already talked about back in the virus chapter. So there's not going to be a whole lot of new stuff here. In fact, a lot of this should be review. One thing that we remember from the virus chapter is that when you compare viral genomes to bacterial cell genomes or even eukaryotic cell genomes, that they are much smaller in terms of the number of genes. And does anybody remember what the reason for that is? Well, you should know the answer to that. I'm not going to answer that now. You should go back and look at that. So when a virus takes control of a cell, and again, we're going to talk about viruses that impact eukaryotic animal cells like what we're composed of, but the argument could be made of really any kind of a cell. The virus, in essence, takes over control of the host cell, doesn't it? And the result is often the assembly and production of viral nucleic acid and protein, and that host cell ends up packaging the nucleic acid, be it RNA or DNA, into a protein coat, and then ultimately the cell will release that virus, which will in turn go on to parasitize, in some respects, future host cells, and the infection continues. Another option, if we think back to that chapter, besides the lytic cycle, is the lysogenic cycle, whereby that viral DNA may become incorporated into the host DNA in the form of a prophage and could sit dormant in that host cell for an extended period of time before it kicks into the lytic cycle and the virus is basically escaping then from the cell, and the infection would start up again. When we talk about DNA viruses and the replication of that viral DNA in a host cell, in an animal cell, we would talk about that process, that DNA replication is taking place in the nucleus of the host cell. In the same way that when a non-infected eukaryotic cell gets ready to divide in the S phase of interphase, that replication process that we talked about earlier would occur within its nucleus. So just remember, DNA replication always takes place within the nucleus of a host cell, whether that's a natural process or whether it is a replication as induced by a viral infection. If we're talking about RNA viruses, then the replication of the viral RNA is going to take place in the cytoplasm of the cell. And that's what the third bullet here kind of talks a little bit about. The synthesis of capsids, which you remember from the earlier chapter, are basically the protein subunits, which when put together form this outer shell within which is the nucleic acid. Remember from earlier in this chapter that translation of protein, or translation of the messenger RNA that results in the formation of the protein, be that in a non-infected cell or in this case in an infected cell, all protein is produced by cells, by eukaryotic cells in the cytoplasm, because you need the ribosome, don't you? Remember, the ribosome is made up of rRNA, and as we described again earlier, in concert with the messenger RNA and the transfer RNA, the result is the formation of the polypeptide. So let's talk about and review a little bit about how this process occurs in double-stranded DNA viruses. So we will assume that the DNA of the virus has gotten into the host cell. That viral DNA will make its way into the nucleus, as we're seeing here as number 1. It's going to pass through the pores on the surface of the nuclear envelope, get into the nucleus. And from here, several things can happen. One thing that can occur is that double-stranded DNA, viral DNA, can be transcribed into viral messenger RNA. Again, the cell is providing the RNA nucleotides and is simply reading one of the 2 parent strands of the viral DNA to make that viral messenger RNA. That, we said, messenger RNA produced in the nucleus will eventually make its way out into the cytoplasm. And as we said a few moments ago, here is where that viral protein will be manufactured by the host cell. The host cell will provide the ribosomes. The host cell will provide the transfer RNA. The host cell will provide the amino acids. The host cell provides everything. All the virus does is give it the directions, the instruction booklet to make the viral proteins. Okay, those viral proteins will eventually come back into the nucleus, and there we will package new viral particles. Again, if this is viral, a DNA virus, then another thing that has to happen to the viral DNA, in addition to being transcribed and eventually translated, is it has to be replicated, right? We have to make more viral DNA. So guess who provides the DNA nucleotides to replicate the viral DNA? You know the answer to that. It's the host cell. And so here we have replication of the viral DNA. It gets packaged again in those proteins. And here's our mature virus particles, which will eventually make their way out of the nucleus into the cytoplasm, and then eventually at some point will be released by the cell. Another thing that can happen, though, remember, is that replicated viral DNA that we have here is number 6, can sometimes be incorporated into the genome of the eukaryotic cell in the form of a prophage. Remember, I was talking about that. Again, go back and review that. And that prophage, that viral DNA, can sit in the DNA of the host cell for a period of several years, and then perhaps some sort of a trigger could cause that prophage to be copied, and we repeat the process of making more viral particles. So again, just a quick review of double-stranded DNA virus biology. A couple things to keep in mind here is that sometimes that cell that has been exposed to viral DNA, sometimes that cell can undergo a transformation process. It's not quite the same thing as transformation that we talked about a little bit earlier in this chapter, but transformation into a cancer cell. And we'll talk a little bit more about how that happens in just a few moments. But any virus that can potentially induce cancer could cause a normal healthy cell to become a cancerous cell. We refer to these as oncogenic viruses. Onco refers to cancer. An oncologist is a cancer specialist, right? You've all heard of that term. Oncology is the study of cancer. So when we talk about viruses like hepatitis B, some of the herpes viruses, the papilloma viruses that you've heard about, sometimes parents will have their adolescent children inoculated or vaccinated, I should say, for papillomavirus to remove the possibility of certain cancers forming when the kids become sexually active. These all are potential initiators of cancers, these viruses that we've just mentioned. And so how a cell becomes cancerous, turns cancerous, is a result of this oncogenic transformation that typically involves viral genes that have got incorporated again into the host cell chromosome as prophages. And if you think back to ANP1, again, we talked about cell division. We talked about the regulatory mechanisms that are normally at work in our cells, such that when we get a cut, that wound repairs itself, right? So there has to be cell division to seal up the cut in the skin and to repair any possible damage that might have occurred, or a fractured bone, right? Repairs itself. That active cell division is initiated and well regulated and orchestrated until the repair is done. So within the cell cycle, as we've said, there are regulatory mechanisms, there are checkpoints. And again, go back, review this. In the case of oncogenic transformation, those checkpoints are not well regulated. Instead of being given red lights to stop, let's say, cell division within a certain stage of mitosis, those particular cells, in this case, get green lights because viral genes are activating, circumventing the normal regulatory mechanisms that would normally occur in the host cell. So the host cell is taken over by the genome, viral genes are expressed, the normal regulatory mechanism no longer is at work, and cancer is the result. So I think it's, again, worth stopping to consider the fact that sometimes simply exposure to certain types of viruses can predispose all of us, potentially, to developing certain kinds of cancers down the road. The last topic that I want to talk a little bit about is described at the very end of chapter 9. It's that section entitled RNA viruses with reverse transcriptase, retroviruses. These are really fascinating viruses, and I'll simply mention HIV, or the AIDS virus, is the classic example of a retrovirus. There are other types, but this is the one you should think of. This is an RNA virus that gets into the cell, and so here we see the retrovirus with its glycoprotein spikes combining with receptors on the surface of the host cell. Here comes the nucleocapsid, which eventually uncoats itself, right? Same old stuff we talked about back in the virus chapter. And in the very center of this retrovirus is RNA. And it's obviously spilling out of the decomposing capsid here. Here's our viral RNA. So what retroviruses do is they reverse the normal process of transcription. Now, you've already learned about transcription. Transcription normally precedes DNA forms messenger RNA. That's transcription. What reverse transcriptase does, and what is that, by the way? It's an enzyme, isn't it? Because it ends in ASE. This is an enzyme that the virus carries with it, and it reverses the transcription process. So now we're going from RNA to double-stranded DNA. That can only happen with the assistance of this enzyme. So here's our viral double-stranded molecule. We call it a provirus DNA is the name that they give to that. And that provirus DNA then goes into the nucleus of the host cell, inserts itself into the chromosome, and it's sort of like a prophage, I guess, in that sense, having incorporated into the chromosome, but they call it a provirus in the case of retroviruses. And there, it can be normally transcribed by the cell. Okay, so here's the cell now making its own messenger RNA, but it's using the viral template, one of these two double-stranded DNA strands, right? And then that messenger RNA leaves the nucleus, goes out into the cytoplasm, and from there we can translate that, of course, into proteins. Not only can we translate it into proteins, but we can also take some of that RNA and package it within the protein. So you're in essence making another nucleocapsid, the very same type as entered the cell, right? It's got the outer protein produced by the host cell. It's got the viral RNA also produced by the host cell. And here we see it, of course, budding, forming this outer capsule envelope that is got these studded glycoproteins. Again, we've talked about all this. And there's our new HIV. So the host cell is not necessarily dying as a result of this budding process, but it has certainly been taken over. And this reverse transcriptase, again, is kind of what runs the show. The other thing that's sort of worth mentioning is the fact that in the case of AIDS, people can have the virus in their cells. It can be in this proviral form for many, many years. And sometimes the person can get sick, seemingly get over the infection, but this can lie dormant for quite a long time. So that's one of the insidious aspects of some of these retroviruses. Okay, that pretty much wraps up all of chapter 9. So this was the third of 3 lectures for chapter 9.